Hi, my name is Cosmo, and this is my STAR project, the Microbial Electrolysis Cell for Sustainable Ammonia Production. So first we have to look at why this is important, and we look at engineering, Earth, and us. So during the last century, we've had relaxed environmental concerns that have influenced Earth as we know it, which shows up in climate change, things like resource caps, growing populations, and other projections that we should be a little concerned about. So moving forward, we look at agriculture specifically and how ammonia is vital for this, as it is a fertilization agent and thus created modern agriculture as we know it. Currently, we use the Haber-Bosch process to create ammonia, which does produce 150 million tons of ammonia per year efficiently. However, it operates at high energy parameters with non-renewable feedstocks, which is not so great for the environment. And this comprises 1.2% of the anthropogenic carbon footprint, excluding transport emissions. So this is something that we're going to have to look at in the future. Right now, we are proposing a new system called the N-fixation microbial electrolysis cell, which has been explored in prior studies, but not in this depth. So we're going to look at the anaerobic anode with exoelectrogenic species to produce protons and electrons. That just means a microbe that can interact with an electrode metabolically, and a proton exchange membrane to create a gradient for those protons to migrate to the cathode side, as well as an external voltage supply, which any MEC would require. And then we're going to pair that with a cathodic N-fixation mechanism, which is the key to this design proposal and literature review that we conducted over this STAR cycle. So our research direction kind of follows that we'll develop an ambiently functional and renewable energy-based NF-MEC for an on-site ammonia production, which will cut down on transportation as well as energy intake. But the challenge is that no microbe exists that can both fix nitrogen and function to interact with the electrodes metabolically. So this is our design premise. So here are some options. The first includes a cathode which would feature a consortia of exoelectrogenic species as well as N-fixing species, kind of combined in some sort of polymeric substance that allows for this metabolic pathway to produce ammonia. And then the next design we're looking at is an open community of N-fixers who would intake hydrogen that has been reduced at the cathode, which is very easy to catalyze and would be very easy to implement. However, the system requires integration that needs a little bit more exploration in terms of the metabolic pathways and microbe interactions. So here are some microbes that we would be considering for these processes, labeled by whether they're aerobic or anaerobic, as well as their main function. So next we have the break-even mathematical model, which follows that using a renewable energy source that's about 15% efficient and at 3 arbitrarily selected applied voltages, and being selective, which means choosing between certain cathodic reactions if the system prefers one or the other. At certain voltages and selectivities, what are the N-fixation rates we're finding, and how can we break even with the natural N-fixation rates so as to create a system that would be improving modern agriculture, as well as comparing to the Haber-Bosch process in ammonia production. So this basically brings us to this intersection graph where we see the 3 selectivities at these voltages that would be required to break even with nature. And we're looking at about 0.8 at the lowest voltage and about 3%, 0.8% and then 3% at the highest voltage selectivity towards ammonia production, where our system would be viable enough to compare to nature and compare to the Haber-Bosch process if it was to ever be implemented locally. So now we're going to finally look at some existing Haber-Bosch process alternatives. These are the ones that I have found in literature that closely compare to what we have been working on and that have helped me develop my design as well. So we have things like artificial root nodules, which are revolutionary, different microbial electrolysis cells, things like that. And they do range in order just because they are different systems and they're in very early stages of development. So we are seeing a lot of experimentation in this field at the time. And our goals that I've seen across the board is to couple this anode with this cathodic N-fixation mechanism in hopes of sparking this new ammonia production. We want to simulate that non-existent microbe, and we want to optimize to eventually break even with nature as well as industry and create this viable alternative. So some experimental questions we might be looking at are using these microbes, these substrates, And under these conditions, what are the rates, what are the selectivities towards N fixation, and what kind of biofilm-solvent interaction are we witnessing? And then a few other side questions that would help us understand the system more. Can microbes eventually be manipulated? Is there a more sustainable and cost-efficient way to produce microbe communities, platinum electrodes, electricity for external voltage supply, and of course ammonia? Is there any other research direction that we should be looking at that might be a little bit simpler, things like that. So in conclusion, we are just hoping to create a system that is more environmentally friendly, more renewable, and more sustainable for a growing population. And finally, I just want to say thank you, especially to my mentor who's been guiding me through this process and being incredibly patient and having an amazing vision for sustainability in her lab, which is Tang Lab. And then I want to thank the viewers who could have napped but instead chose to listen to me talk about microbes. And I also want to thank Drexel University, obviously, and the OUR staff for offering me these opportunities and for being very accommodating during these strange times.